Hello children, in the last lecture we have been discussing regarding the amino acids, right? So in amino acids we have seen different kind of amino acid, essential, non-essential, and even we have discussed with the standard, non-standard amino acids, and later I have discussed with the structures of amino acid, the different categories of amino acids, and in this lecture we'll be mainly discussing with the last classification of the amino acids. So what are the type of classifications that we have learned under this chapter? So one, essentially the most important category for the amino acids, is being the categorization of amino acids into essential and non-essential category. So there we have learned 10 amino acids are coming under essential and other 10 will be under the non-essential category. And later I have given a classification based upon their structure, the chemical structures we have learned. So type of amino acids falling under different categories that we have discussed, like you can say the aromatic amino acids, the simple or neutral amino acids, we have sulfur containing amino acids, and even we have discussed regarding the basic amino acids, acidic amino acids, and heterocyclic amino acids. All these different categories we have discussed in the last lecture. Along with that we have discussed with the polypeptide formation. What is a bond that is generally present in the amino acids, children? It is the peptide bond. Peptide bond, it is nothing but the chemical bond which is formed between the two amino acids. Any of the amino acids, they will be joined with the help of the peptide bond. And these peptide bond, essentially the peptide bond means it is the bonding between the carbon in the carbon of one amino acid to the nitrogen of other amino acid. It is nothing but the acidic group in one amino acid, it will be reacting with the basic group of other amino acid. When they meet together, they will be forming a peptide bond. Okay, so this is regarding the last lectures we have discussed regarding the amino acids, and even we have discussed with certain important things like the directionality. I have told you always in reading or writing an amino acids, group of amino acids, we always follow this N2C rule. Always will be starting with the N and will be ending with the C. Amino acids towards the N terminal region is said to be the first one, and towards the end of the C terminus will be the last amino acid. Okay, so this is what the important points we have learned regarding the amino acids, and in that mainly I was talking with the classification system. So classification, the two classifications I have told you, one basing upon the chemical nature and another it is based upon the production inside the body, essential and non-essential, and some are semi-essential we have discussed. And the last important classification which is very important for you, that is regarding the polar and non-polar amino acids. Some of the amino acids, they are said to be polar again. Okay, so different type of amino acids, they are grouped under the polar amino acid group. Why? Because mainly they will be forming the hydrogen bonds with the water. We all know polar means the chemical molecules in turn, they are highly reactive with water, which are said to be the hydrophilic. Even in the plasma membrane, in plasma membrane, the cell chapter, even I have discussed with this hydrophilic and hydrophobic. Hydrophilic is nothing but water loving substances, and hydrophobic is the water hating substance. Hydrophobic is generally the hydrocarbon chemical molecules, they will be showing the hydrophobic nature. And hydrophilic molecules are the ones which are highly reactive with water. They will be having different type of groups which have the ability or tendency to bond with the water through hydrogen bonding. So in the same way, there are certain amino acids which will be highly reactive with the water, and they are said to be the polar amino acids. As they are easily dissolved inside the water, they are said to be the hydrophilic. Okay, so here I have given the list of amino acids. We have 9 amino acids in this group here. So all these 9 amino acids will be coming under the polar amino acid group or hydrophilic amino acids. and there are the remaining amino acids, all the 11 remaining amino acids. So out of 20 we are discussing, we are not interested in learning the non-standard amino acids. We are always interested in learning the 20 general or standard amino acids. So there the 20, the 9 will be coming under the polar category and other 11 will be coming under the non-polar category. Non-polar means they are less reactive with water, or generally they have the water hating molecules and hence they cannot be dissolved inside the water, which in turn means they cannot form hydrogen bonds with water. And such type of amino acids are said to be the non-polar amino acids. Okay, so non-polar can also be called as hydrophobic. Polar are said to be hydrophilic. The non-polar are said to be the hydrophobic amino acids. Okay, so today we'll be first starting with the last classification of the amino acids, that is polar amino acids or hydrophilic, non-polar amino acids or hydrophobic amino acids. So this is what we'll be discussing in this lecture, and later I will be discussing with the structure of the proteins, and even we'll be trying to finish the functions of the protein, if not in the next lecture. Okay, so this is regarding the topic today, children. So first we'll be starting with the hydrophobic and hydrophilic amino acids. So this is one of the classification of the amino acids based upon their ability to react with water. Okay, so mainly the water, the 70 to 75% of our body, it is mainly made up of water. So all the chemical molecules, the macro, say micromolecules, they must be having some interaction with water. Either they are hydrophilic or they may be hydrophobic. Hydrophilic means they will be dissolved inside the water easily. Hydrophobic means they will not be dissolved inside the water easily. So all the macro or micromolecules, mainly they are categorized under the two groups, and hence it is important to learn about the amino acids which will have the ability to dissolve in water and the amino acid which will not. Okay, so first let us see the two different categories. Here again you have to remember all the names of the amino acids, and hence I have made some mnemonics here. I have asked you to learn all these amino acids in the last lecture, children. So it is very important. So before going to learn the classifications of the amino acid, first you have to understand and learn the all 20 amino acids. Then only it is very easy for you to classify or easy for you to remember all these classifications. So before going to learn all these classifications, don't be in a hurry to learn all the classification. First you take some time to learn all the 20 amino acids. So when the terms are very familiar to you, then you start learning all these classifications. So three classifications mainly I have given: essential, non-essential. We have discussed with the amino acids which are having different type of chemical groups, and the last classification I have told you regarding the hydrophilic, hydrophobic, or you can call it as polar and non-polar. Okay, so here again we have the classification. Here we have the 9 amino acids and here I have the 11 amino acids. So if you see all this category, let us read it out here: asparagine, cysteine, glutamine, threonine, glutamic acid, aspartic acid, lysine, arginine, histidine and serine. So all these 11 amino acids will come under the non-polar category, which means they are less reactive or non-reactive with water. Hydrophobic, generally they have hydrocarbons, more number of hydrogen and carbon atoms, which will make them repel with the water. Okay, so look at this. So what is the mnemonic which I have made here? Aishwarya Rai, the Bollywood actress. So on her we are making a short statement for this. Okay, so look at this statement which I have given. So many people are admired by her beauty. So based upon that I have made a mnemonic. Okay, so here if you see the mnemonic: ash, celebrity, glamour, thunderstruck, typical glam aspirants lying around Himachal state. So mainly I am saying that there are a group of people, typical people in Himachal state, who are amazed. They are very much shocked or amazed looking at the celebrity glamour of the Aishwarya Rai. So that is what the mnemonic is here. Okay, so that is what the mnemonic says. And if you see how you will compare this with the amino acids, let us see here. Okay, so asparagine, AS. So here it will be matching with the Aishwarya Rai. In short, I am calling it as ash. Okay, So ash, cysteine, for cysteine I am giving celebrity, okay, celebrity and cysteine. We don't have any amino acid starting with C, so simply C means always cysteine, okay. So celebrity means cysteine. And next we have glamour. Glamour here is for glutamine. So first two letters GL, it is for glamour. And next we have threonine, TH. So thunderstruck. Thunderstruck means amazed or shocked. Okay. So next we have tyrosine starting with TY, and hence I have given typical. Okay. And glutamic acid GL, and here I have given glam, where here also you have the first two letters which are GL. And next we have aspartic acid. Here I have given aspirants, glam aspirants, the people who are interested in who are admired with beauty, they are said to be the glam aspirants. And next we have lysine, ly, lying, arginine, around AR. And next we have histidine, HI, that I have given as Himachal, okay. So state Himachal Pradesh in India, we all know. So HI, Himachal, that is histidine. And lastly we have serine, that is starting with the S. We don't have any amino acid starting with the S, so we don't have any problem. So the first letter I have given as the S here. Okay, so I have not matched two letters here, I have matched only one letter starting with the S. And also for, I have told you, the cysteine, we have only one type of amino acid which is starting with C. So simply I have given the first letter C, celebrity, not the two letters I have considered where I have matched. If you compare with the other amino acids where I have mainly matched with the first two letters, but for cysteine and serine I have matched only the first letter. Okay, so this is about the mnemonic. And once again, listen carefully children. Ash, celebrity, glamour, ash, celebrity, glamour, thunderstruck, typical, glam aspirants. Ash, celebrity, glamour has made typical group, a special group of glam aspirant. Who are the special group here? Glam aspirants. Aspirants means mainly who are admired by beauty. So such type of people who are living around Himachal Pradesh state, they are mainly amazed by the celebrity glamour of Aishwarya Rai. Okay, so once again, so the people living around the Himachal state, in them we have a typical group of people who are glam aspirants. Glam aspirants means who are admired by beauty. Okay, those people, they are thunderstruck, amazed by the celebrity glamour of Aishwarya Rai. Okay, so this is all about the mnemonic. So the mnemonic here, it is completely matching with the type of non-polar amino acids. So what you will do, you will simply learn this mnemonic so that it will be easy for you to remember all the non-polar amino acids. Okay, and always remember children, if you are going on making some codes or mnemonics for all the things which you have to memorize, then you will be landing in confusion. And hence what I request you to do, mainly if you have certain mnemonics in the same place. So let us say I have given a mnemonic for polar as well as non-polar. So what you will do here, in order to provide an option, I have given mnemonics in both the cases, but I want you to learn only one mnemonic, so it will be easy for you. If you are learning with one mnemonic and one group of amino acid, it will be easy for you to understand, or which will imply the remaining amino acids will be coming under the other group. So it is not important to learn the mnemonic of the other category. So it is very simple if you learn any one of. So I am giving option for you. If this mnemonic, it is easy for you, you start learning with the polar amino acids and with that mnemonic, and you leave this mnemonic as well as the amino acids. So whenever there is a question regarding the non-polar, first you have to remember what are polar, and that will help you remember about the non-polar amino acids. So in this way it will be very easy. In order to give you an option, I am making mnemonics, but if required, whenever it is required, then only you have to remember certain terms with the mnemonics. Otherwise, you should never use the codes. Always try to memorize the things. Next, so coming to the second type of amino acid group, that is hydrophilic or polar amino acids here. So in this polar amino acid group, let us see the list of amino acids falling under this category. We have the methionine, proline, glycine, alanine, phenylalanine, tryptophan, leucine, isoleucine, and finally we have valine. Okay, so for this, a small story. There is a mother who loves their children. All the mothers, they generally love their children. The mother prays God. At the same time, she asks her progeny. Progeny means children, to live in victory. So she desires her children or progeny to live always in victory. Okay, so she is praying on one side to the God, and she is asking her children to live in victory. She is blessing them. Okay, so that is what about the mnemonic here. So if you look at the mnemonic, methionine matches with the mother. Okay, proline matches with praise, glycine with God, alanine with asking, phenylalanine with progeny, tryptophan with to, leucine with live, isoleucine with I in, valine with victory. V and V. Okay, so mainly here I have matched only the first letter. Why? Because we have given a clear demarcation for the non-polar group, and hence I have matched only the first letter here. So this is regarding the list of amino acids under polar category. So if you see here, the mnemonic says, mother prays God asking progeny to live in victory. Okay, even you can think that she is praying God in that way, or you can think that she is blessing her children to live in victory. Okay, so mother prays God asking progeny to live in victory. Okay, so this is one of the mnemonic I have made for the polar group of amino acids. Okay, so this is regarding the two group children, the amino acids coming under the polar and non-polar category. So polar, for polar, what is the mnemonic I have given here? Mother prays God asking progeny to live in victory. Okay, and regarding the non-polar amino acids, it is little lengthy. Ash, celebrity, glamour, thunderstruck, typical, glam aspirants lying around Himachal state. Okay, so this is regarding the two mnemonics. So once you have done with all the 20 amino acids, slowly start learning the essential and non-essential category. And later what you will learn with the chemical groups which are mainly present in the amino acid. Extra groups, like we all know, all the three functional groups are common to the amino acids, and even for the simple amino acids we have the same thing, but we have certain amino acids where the additional functional groups are present, and that you will learn. And lastly, you will learn the polar and non-polar amino acids. Okay, even you can discuss with any kind of doubt. Even we have some charged, uncharged amino acid, negatively charged, positively charged. That is not important here or required here. If you have any doubts regarding that, you can post me or you can comment me, so that I will be helping you in this. And next, we'll be discussing with the protein now. So till now we have discussed with the amino acids, their importance in the formation of the protein, the peptide bond formation, type of proteins, dipeptides, polypeptides, oligopeptides and macropeptides, and regarding the classification of the amino acids I have given: essential, semi-essential and non-essential. We have polar, non-polar, and also the structures of the amino acid. And lastly we have the proteins. So now we have learned with the basic units or monomers, you can say the building blocks of the proteins regarding amino acids. Now we'll be studying with the proteins. So all the amino acids, they are joined together with peptide bonds to form the macromolecules, which are proteins. Proteins are generally polypeptide. So the linear chain which I have drawn to show you the structure of a single peptide a single polypeptide. For a protein molecule, generally many molecules of polypeptides are present and they are jointed to form a single protein, and such type of proteins which are having more than 100 amino acids or more, we call them as a macropeptide. Okay, so now we'll be learning with the proteins. So mainly in the case of the proteins will be generally, all the proteins will be containing all the general or standard amino acids. So all the proteins will be made up of the 20 amino acids. This is a general condition. Yes, we have certain conditions where some of the proteins, or rarely you can see, some of the amino acids are missing in certain proteins. For example, if you take the cereals, the grains, they are mainly rich in the case of methionine, but they are deficient in the lysine. So this is one example here. And even when you compare with the pulses, the reverse condition will be there. They are rich in lysine, but they lack this methionine. So there are certain proteins, generally one or two amino acids, they are insufficiently present, or you can say they are generally absent. So in order to satisfy the needs of our body, mainly with the essential amino acids, always we have to be very keen in having this amino acid, the proteins, in consuming the food which are mainly containing the essential amino acids. Okay, mainly the staple food of India, we all know that is mainly the rice and wheat. So if you compare the cereals are generally rich in the methionine, but they lack the lysine. So what you will do, you have to supplement this lysine from other proteins. So what are the other proteins I have told you? The pulses. The pulses will be having more of lysine and less of methionine. So when you have this combination of foods, then it will satisfy the needs of our body, mainly with the essential amino acids. But generally all the proteins will be containing all the 20 amino acids. Okay, so now, so in this protein, sometimes some proteins will lack the amino acids, some are generally meager in amount or less in amount. So in order to balance all the things, we have to consume different type of combination food, so that we will be having the sufficient amount of essential amino acids. Okay, so this is regarding your proteins, the macropeptides made up of the peptide bond. So here I have given children, proteins are large sized macromolecules. They are having one or more polypeptide chains here. So one polypeptide chain, it's a linear chain. Generally we represent the linear chain of polypeptide in this manner. In this way, many polypeptide chains are linked together to form a single structure of the protein. Okay, now let us see how these proteins are associated to form, how these amino acids are associated to form the functional proteins. Okay, so if the free amino acids are present, they will be having, they doesn't have any role. Okay, so free amino acids are rarely used by the body. All these amino acids, when they are jointed together to form the complete structure of the protein, which is biologically active. So we'll be discussing with the structure of the protein now. And the most important thing here you have to remember regarding your NEET, it is even mentioned in your NCERT book, the most abundant protein in animals or in animal kingdom is collagen. Okay, so what is the important protein which is present in animal kingdom, children? Collagen. And the most abundant plant protein which is present in excessive amount in the plant kingdom or in plants is the enzyme rubisco. It's also a protein and hence it is said to be the important one. So both are important here. One is rubisco for plant kingdom. Even I have mentioned this in the last lecture, children. So any extra information you have gathered with any of the chapter, it has to be started from your NCERT book. First you have to be thorough with the NCERT book, then only you will add some extra information with that particular subject. Okay, so regarding the structure of the protein we'll be discussing now. Generally in biology, the protein structure, it is mainly discussed in 4 steps. 4 steps means mainly the biologist, they will be explaining the structure of the proteins in 4 different ways. Okay, so what are the 4 different ways that we will learn? Generally, the proteins have 4 different conformation. 1, the primary structure, 2nd, secondary structure, tertiary structure, and quaternary structure. So here I have mentioned all the names. So what is the classification for the proteins based upon their structure, children? Primary, secondary, tertiary, and quaternary structure. Now let us see how these structures are formed and how they are present and what will make them biologically active compounds. Okay. So first we'll be discussing with the very basic primary structure of the protein. Primary structure means it is nothing but it is our imagination. It is simply for our understanding we are mainly giving this primary structure of the protein, but there is no experimental evidence till date regarding the primary structure of the protein. We all know the proteins are synthesized through a process called translation. So inside the nucleus we have the DNA. DNA will be releasing its message and that will be read by the ribosome and it is converted into the protein in the cytoplasm for eukaryotes. For prokaryotes there is no distinction between the cytoplasm and nucleoplasm. Why? Because nucleus is absent there, the cytoplasm will be showing both the reaction, transcription as well as translation. Okay. So here the protein synthesis will be taking place for the formation of different kind of proteins and the message it is derived from the DNA present in the organism. So here proteins, when they are formed, the first basic step of the protein synthesis, that is the translation process. We all know there is a linear chain, many amino acids, they are joined together to form a chain like structure. So this is completely hypothetical. We don't know whether the protein it is a straight chain. Why? Because till date we don't have any experimental evidence. We are simply thinking or imagining that there is a protein which can lie in a linear chain. But till date it is not proved. Okay, so simply we call it as a hypothetical structure. So what is the first structure or the primary structure of the protein? When all the amino acids are joined together with the help of a peptide bond. We all know there is a peptide bond which is a covalent bond. So what is a peptide bond, children? It's a covalent bond. It's the strongest bond. This peptide bond will bond all these or will bind all these amino acids together. So with the help of this covalent peptide bond, all the amino acids will join and they will form a linear chain here. So in the primary structure of the protein, only one chemical bond is seen, that is the peptide bond. Let us write it here. And what I have told you, children, I have mentioned that the peptide bond is the strongest covalent bond. Covalent bond is formed by the sharing of the electrons, we all know. So it is the strongest bond and the peptide bond is existing in this linear chain. Remember, the proteins will never be present as a linear chain molecules. It is hypothetical. And even I have mentioned the direction, that is N to C, you have to remember. So this is regarding the primary structure of the protein. If the proteins are simply, if we are talking with a single chain, the primary structure can be present like this. Okay, so they all are present attached with the peptide bonds and they are linked together. And what about the secondary structure? So generally you can see the secondary structure of the proteins from starting from the secondary till quaternary structure. These are real structures. Why? Because we have experimental evidence for these structures. So let us learn with the secondary structure of the proteins here. So here in this case, the secondary structure of the protein, it can exist as alpha helix. It is called as alpha helix. Remember, the second one is said to be the beta pleated sheet or beta motif. So what are the names, children? The secondary structure of the protein, it can be the alpha helix. What is the second type I have told you? The beta pleated sheets or beta motifs. Okay. So these are the two types of secondary structures of the proteins. They are formed in certain enzymes or molecules, and the formation of the proteins you can see this structure. How these structures are mainly present? This alpha helix, so mainly discussing with the alpha helix. Alpha helix is simply a curled ribbon. It is just like a curled ribbon. So look at this structure, children. Okay, so this is nothing but a spiral. It's a curled ribbon like structure. So alpha helix will be present like this. It's a curled ribbon. So here I have given the structure. So this can be left-handed curl or it can be right-handed curl. So right-handed curl, it will be generally, we say the parallel or anti-parallel nature. So the alpha helix can be present as right-handed or left-handed helix, which is parallel and anti-parallel. So this is how generally the alpha helix will be present. Okay, so it is helical and it is like a curled ribbon. Okay, and coming to the beta pleated sheet. How is a beta pleated sheet? So it is just like a pleating. We all know the pleats are made for different type of fabrics. You can see the pleats. So even for your fabrics, you can see the pleating. So here if you observe, this is a pleated structure. Look at the structure, children. So if you take a ribbon and when you fold it, when you take a thick ribbon, when you fold it and later when you open, it will give a structure like this, and this is said to be the pleated configuration. Okay, so this is the structure of the protein, which is the beta pleated sheet, or you can call it as a beta motif. Okay, so simply if you observe, as we cannot see the three-dimensional view of the structure, simply I have made with some color papers. So look at the structure, children. The secondary structure of protein can be alpha helix, which is simply a spring, or you can say the curled ribbon like structure, and beta pleated sheet, it is just like a folded ribbon, which is a thick ribbon. Okay, this alpha helix is it's like a thin ribbon. The beta pleated is a thick ribbon like structure. Okay, this is folded and pleated. Next, so this is regarding the secondary structure of protein. So now let us see what type of bonds are present in the structure. There are different type of chemical bonds. So other than peptide bond, we have different type of chemical interactions present in these proteins. All these amino acids, due to their negatively charged, positively charged, or you can say hydrophobic, hydrophilic, different type of chemical nature is present for different type of amino acid. Based upon that, they will be showing different type of links or chemical interactions. Other than the primary chemical interaction for any protein is the peptide bond, right? So if a protein is formed, it is generally formed with amino acids, and amino acids are linked by the peptide bond. How this peptide bond is formed? Between carboxylic group of one amino acid to the amino group of the other amino acid. The C-An link is said to be the peptide bond. This is a dehydration reaction. In formation of one peptide bond, it will release one water molecule. Dehydration or condensation reaction, simply like that of the glycosidic bond. If you remember, there I have told you it's a dehydration and condensation reaction. The same reaction will be taking place. For every peptide bond formation, one water molecule will be released. Dehydration. And for every peptide bond formation, ATP is required. Even it is energy expensive process. Okay, that you have to remember. And next, coming to the alpha helix. So now let us say we have a straight chain and we will slowly twist it and it will become a curl like this. It's a ribbon or a curl, and that is nothing but a alpha helix. So generally the proteins are present in this manner. Why? Because we have experimental evidence for this. So here in this alpha helix, what type of interactions you can see? So we have the amino acids like this, children. So I have given a twisted ribbon or curled ribbon like structure over which I have placed the circles, which means amino acids. So look at this carefully. I have given a curled structure over which all the amino acids are arranged. So when they are placed together, we have some kind of chemical interactions between them. And the first important common interaction that has to be present for any structure is the peptide bond. First we will write it here. Okay. For the linear chain, we have only one peptide bond, that is okay. So here the peptide bond is only bond present for the primary structure. What about the secondary structure? Peptide bond is always present. First we will write the peptide First way I have written the peptide bond. What is the second type of bond which is seen here? First we will discuss with the alpha helix. In alpha helix, generally the amino acids will be forming the hydrogen bonds, where these hydrogen bonds are formed. The hydrogen bonds are formed between the amino acids. The hydrogen of one amino acid to the nitrogen and oxygen containing amino acids are present generally. All the 20 amino acids are generally seen in the proteins. So what will happen, children? The oxygen and nitrogen it is common. Oxygen, or you can see the nitrogen, the more electronegative atoms wherever they are present, they are linked with the hydrogen of the other amino acid. So simply if you see here, wherever there is a close interaction of the amino acid, there you can see the formation of the hydrogen bond, which will make this structure more stable. So for its stability, other than the peptide bond, the alpha helix will be having the intramolecular hydrogen bond. So there is only a single chain. You have to remember I am talking with a single chain. Inside this chain itself, the molecules or the amino acids are having the hydrogen bonds, and such type of hydrogen bonding is said to be intramolecular hydrogen bonding. Intra means within itself or within inside the structure. It is said to be the intra. In between two layers or in between two peptides, it is said to be the inter. Inter means in between, intra means within, or you can say inside. Okay, so here what type of bonds are seen here? One, we have the peptide bonds, and the second we have the hydrogen bonds. And there is another bond, there is even other bond which is called as the disulfide bond. What it is known as? The disulfide bond. So what are these disulfide bonds here? So I am discussing with the third type of interaction present in the alpha helix. One I have discussed the peptide bond, which is common. Second is hydrogen bond. In hydrogen again we have intramolecular hydrogen bond. And third I am discussing with the disulfide bond. So pay attention, children. It is very, very important to learn these structures, and it is very difficult to understand. So when you follow me in a proper manner, it will be very easy for you. Okay, so look at this. So here we are talking with the third important bond, which is a disulfide bond. How this disulfide bond is formed? Disulfide bond, it is generally formed between two cysteine residues. What I am saying? The disulfide bond, it is formed between two cysteine residues. Two cysteine means cysteine is a kind of amino acid. So wherever we have interaction of two cysteine molecules. So let us say in this spiral or in this curved structure, we have one cysteine here and another cysteine which is placed here. They are in close proximity. So when two such cysteine molecules are in close proximity, immediately they will form this disulfide bond. So disulfide bond, mainly this is also releasing a water molecule. So two cysteine molecules, if you remember the structure of cysteine which I have given in the last lecture, there you can see it is made up of the sulfur molecules. The sulfur is present in cysteine amino acid. So when two cysteine molecules they come together, these sulfur molecules, they will release their hydrogen in the form of water, and they will be added with the oxygen and they will form this disulfide bond. It is not required here the reaction, the chemical reaction. Simply understand the oxidation reaction will cause, with the addition of the oxygen, they will be forming the disulfide bond. If there is reduction reaction, addition of hydrogen, it will lead to the breakage of the disulfide bond. Okay, bond making and bond breaking. It will break with the reduction, it will be made with the oxidation reaction. So this is how the disulfide bond is created. It is created by the close proximity or interactions of the cysteine amino acids in the protein chains. Okay. Now, so all these three are present for the alpha helix. So in this, if you see, in these three bonds, the sulfur, the disulfide bond and the peptide bond, both are the covalent bond. So what do you mean by covalent bond? It is the strongest bond compared to non-covalent. It is the strongest bond. So the covalent interactions are 1 the peptide bond and 2 is the disulfide bond, and hydrogen bond it is coming under the non-covalent interaction. Okay, so these are loosely attached bonds here. Next, coming to the beta pleated sheets. So just like pleats, they are present like pleats. This is like spiral. And in these beta pleated sheets here, how these thick structures, why I am calling it as a thick ribbon? It's a slim ribbon. Alpha helix, it is nothing but a ribbon or spirally coiled ribbon, and here it is a pleated ribbon. Because of its thick structure, it is being pleated. It is not twisted or turned in a regular fashion. So why these are pleated here? Because they are forming the thick structures. How these thick structures are made, that we will see. So here I am mainly showing you the beta pleat formation, how these thick structures are formed. Other than the spirals which are thin, here these are thick structures, how these are formed. So look at the structure. So this line represents, this is a single strand and it is turned and it is again forming another strand. Again it is folded back to form another strand. So in this way, here I am mainly showing you 1, 2, 3, 4 and 5 strands here. Let us mark. The first 2 strands are present here. Third strand it is empty. Empty means you can say it is single. These are paired here. These 2 are paired and this is said to be the single strand. It is not paired, non-paired strand. And here we will mark it as 4 and 5. So total we have 5 strands. The first 2 are the pair which I have represented and 4 and 5 are paired here. The 3 it is present in a unpaired fashion. Okay. So let us see. So let us write some lysine, methionine, proline, histidine. Any amino acid you can write, valine, tryptophan, phenylalanine. So randomly I have given some amino acids here. So all these amino acids, they are present. So if it is a spiral, we don't have any close interactions between these amino acids. It is a single spiral. Okay, so this is a single spiral here. So here, in this case, the interactions are present between the amino acids in the single chain. So in this case, I have told you the single chain interactions will be present. In the same manner, a single chain will have the intramolecular hydrogen bonding, and the amino acids on the opposite strands, even they have the interactions, which is said to be the intermolecular hydrogen bonding. Are you getting me, children? We have 2 kinds of interaction here. We have different strands which are formed by folding and twisting, and these foldings are in turn stabilized by the strongest hydrogen bonds. So here even for alpha helix we have the hydrogen bonds, but they are mainly present adjacent to each other. So which are placed closely, they will be having the hydrogen bond formation. But in the case of the beta pleats, the strands, though they are lying in the opposite orientation, the adjacent amino acids will form the hydrogen bonds. Adjacent amino acids forming the hydrogen bonds is said to be the intramolecular hydrogen bond, same like that of the alpha helix. The side by side amino acids, they will show the intramolecular hydrogen bonds, and the amino acids which are present in 2 different strands, when they are twisted, it will become another strand. So when they are present in between the 2 strands, it is said to be the intermolecular hydrogen bonds. So mainly here we have a ladder-like configuration. So in this, the amino acids on either side are linked by hydrogen bond, which is said to be the intermolecular hydrogen bond, and the molecules which are having the adjacent hydrogen bonds, they are said to be the intra-hydrogen bonds. Okay, so we have intramolecular as well as intermolecular hydrogen bonds. Due to this intermolecular hydrogen bonds, this complete 2 strands, they will appear like a single strand. In this spiral case, if you see the spiral, the alpha helix, we have only 1 strand. We don't have any inter-hydrogen bonds here. So that will not give you any thick appearance. But in the case of beta pleated pleated sheets, two strands, they are jointed like a ladder and they will form a thick sheet. So I have shown you the structure before. So look at this structure, children. So here the thick structure, it is mainly formed by the two sides here. So here if you observe, thickness is greater compared to that of the spiral. And how this thickness and pleating it is created, this is only because of the intermolecular hydrogen bonds. Okay. So other than the peptide bond or the disulfide bond, the beta pleated sheets in addition, they will have the intermolecular hydrogen bonds. Clear with this, children? Once again I am telling you, the thickness of the beta pleated motifs or sheets are more compared with that of the alpha helix. Why? Because the alpha helix will only have the adjacent interactions or the formation of hydrogen bonds, which is said to be the intramolecular hydrogen bonds. But in the case of beta pleated sheets, along with the intramolecular hydrogen bonds, we have intermolecular hydrogen bonds. So when this molecule or amino acid chain, when it is twisted or turned many times, some of the gaps, they are linked by the hydrogen bond. So when these hydrogen bonds are formed, it will form a thick structure. So look at this. So here I have given a thick structure. Later I have given a thin structure. Again a thick structure, thin structure, thick, thin, thick, thin. So in this manner, some of the strands are unpaired or they are left as such without pairing, and some of the strands, they will be forming the thick structures. So thick, thin, that will be seen associated. Sometimes all the thick structures can be seen. So based upon the structure of the protein mainly, they will show two type of interaction. So beta pleated sheets are the ones, they will be having the structure which is wavy. Okay, so it will be present like this. Some are unpaired sometimes and some are paired here. The paired condition, it is mainly because of the intermolecular hydrogen bonding. Okay, so let us mention this. So along with all these three, the beta pleated sheets in extra, they will have intermolecular hydrogen bonds. So what are the four bonds that are present for beta pleated sheet? We have intermolecular, intramolecular hydrogen bonds, we have peptide bond in common, and we have the disulfide bond. So this is regarding the structure of the beta pleated sheet. For alpha helix and beta pleated sheets, both are coming under the secondary structure of the protein. Clear with this, children? Simply remember, the alpha helix is a curled ribbon and this is a pleated ribbon which is thick. Okay. So this is regarding the secondary structure of the protein, and here are the interactions. We have the two covalent interactions, that is disulfide and peptide bond, and the other is a non-covalent interaction which is the hydrogen bond. For beta pleated, we have inter as well as intra-hydrogen bonds. That you have to remember. And next, coming to the tertiary structure of the protein. So this is the main tertiary and quaternary structures are the major structures which are mainly, why I have not given any examples for this, children. So these are the individual molecules. So simply one alpha helix or one beta sheet that you can never find in a protein molecule. If a protein molecule is biologically active, the sum total of alpha helix or some total of beta pleats are present, or sometimes the combination, mainly the combination, alpha helix along with the beta pleats are present. So here I am not giving any examples because we don't have any such proteins where only one alpha helix or one beta sheet is present. They are generally present in combination, or they are present as repeated units. Many alpha helix, they will bind together, or many beta pleats, they will be binding together to form a protein molecule. So here coming to the major important ones, the tertiary and the quaternary structure. Why these are important? Because the biologically active proteins are always in this conformation. Either they are tertiary or they are quaternary. Now you will understand. So these two are the basic structures which are representing the units of the proteins. These are forming the units, and these are the actual proteins which are biologically active, or which are mainly the functional proteins. Okay, so look at this. So here if you see the structure, what I have given, children, I have given a combination of alpha helix, and inside I have given a beta pleated one. Okay, so alpha helix together with the beta pleated. And how this is forming, children? It is forming a globe-like or a circular structure. So overall, if you define a tertiary structure of the protein, you can say that the tertiary protein or a tertiary structure of a protein is nothing but the overall conformation or the overall folding of a protein into a single shape. It is said to be the tertiary structure of the protein. Let us say there is two strands here, two alpha helix or two beta pleats, anything. All these structures, they are together forming a ball or wool-like structure, which is giving a globular shape to the protein. It is said to be the tertiary structure. So these individual units, they will bind together, and in combination they will form a wool or wool ball-like structure, which is called as a globular protein. Such structures are said to be the functional molecules, or it is said to be the tertiary structure of the protein. So generally the proteins are in this conformation. So the native state of the proteins will be in the tertiary and quaternary structures. So this is regarding the simple proteins. The simple proteins, they are generally having this conformation. So here I will be giving one example, children. So when all the alpha helix or beta plates, they are arranged in a linear fashion and they will form a rope-like structure, such type of simple proteins are said to be the fibrioids. Okay, so fibrioids means they are nothing but their long chain or rope-like structures. And there are certain proteins which will be binding together, which will be having internal chemical interactions, and finally they will be converted or modulated into a circular globular structures, which are said to be the globular proteins. So this is regarding a single protein molecule I am discussing, children. Single protein molecule having a single polypeptide. So one polypeptide having this structure, a ball-like structure, or one single polypeptide forming a rope-like structure. The rope can be made up of alpha helix and beta plates, and even the globular structure can be made up of alpha helix and beta pleated. So if you see here, both are jointed. Some part of the polypeptide is alpha helix, some part of it is said to be the beta pleated. So both are present and both are together forming a ball-like structure. So I am discussing with a single polypeptide. The tertiary structure means a single polypeptide. Single polypeptide forming a circular protein. This kind of simple protein is said to be the globular protein. Single polypeptide, it is forming a linear rope-like structure. It is not jointed end to end like a circle. It is simply a rope-like structure. Such type of protein is said to be the protein. Okay, they are rod-like structures. Simply will write it here, children. Okay. So this is regarding the tertiary structure of the proteins. So what I am telling you, children, tertiary structure, it is about a single polypeptide. After primary and secondary foldings, these protein chains or the polypeptide, the single polypeptides, they are folded into a shape, and that shape can be a circular shape which will form a globular protein, or it can exist as a rod-like linear shape which is said to be the fibroid protein. So these two are the type of proteins which are generally seen in the formation of the tertiary structures. So here I have given some examples, children. The immunoglobulins, the defensive proteins inside our body, they are generally globular and they are the simple proteins. And the fibroid proteins, the rod-like proteins, the shape of the protein, it is rod-like, which are generally coming under the keratin, the collagen. They are generally the tertiary structure of the proteins. They have the rod shape. Okay, some are fibroid proteins and some are the globular proteins. The tertiary structure of proteins are generally helping in the formation of enzymes. These are mainly associated with the formation of enzymes and also the receptor proteins. I have discussed this in the plasma membrane. We generally have receptors present on the plasma membrane. The receptors are nothing but the proteins. So the proteins will be mainly having this conformation. The receptor proteins and also the enzymes, they generally have the tertiary structure. These are simple proteins. Even we have the complex enzymes, the multi-subunit enzymes, they are generally having the quaternary structure. So next we will be talking on the quaternary structure of the proteins. So look at this. Here we have different type of polypeptides. So here essentially we have only one polypeptide that we have discussed. So some part of it is beta pleated, some part of it is alpha helix, or everything, the complete polypeptide is alpha helix. So the complete protein is beta pleated. It may differ from protein to protein, but this is the association, or this is mainly with the one polypeptide. But in the case of the quaternary structure means we have different polypeptides, 4 or 5 polypeptide. Each polypeptide will have a different configuration. So if you want to know the shape of a quaternary protein, the overall shape, it is dependent upon the individual shape of the molecule. So some irregular shapes will be created. Sometimes the globular shapes may be created. It depends upon the individual polypeptide present in that particular protein. Okay, so look at the structure which I have given here. So here I have given one beta alpha, beta alpha, beta alpha. So each molecule is one polypeptide here. Okay, so how many polypeptides I have given here, children? I have given 4 of them. So all these 4, when they are jointed together, when they are interacting with each other, they will be forming a different structure. Individually they will have one structure, but when they are jointed, when they are interacting with each other, they will be forming a different structure. So here mainly the joining of many more than one polypeptide chain will be taking place. That is known as the quaternary structure. Now, so for this I will be giving a best example, the myoglobin and hemoglobin. The hemoglobin, it is made up of 4 polypeptide chains, and all these 4 polypeptide chains are linked together. In the same way we have myoglobin, okay? So muscle protein, myoglobin and hemoglobin, right? So both are examples for quaternary structures. Complex protein molecules. So the basic thing you have to remember here, in this case I have discussed with the type of bonds present. Then tell me, what about the tertiary structure and quaternary structure? Why I haven't discussed this here? Because both the structures will have all the bonds in common. So all the chemical interactions are mainly seen in the quaternary as well as the tertiary structure. Then what extra you will have in comparison to the secondary structure and primary? So in primary, till here I have told you peptide bond present for both. Later we have hydrogen bonds, disulfide bond, inter and intramolecular bond. So if you see here, 3 type of bonds. In hydrogen we have again 2 subtypes, okay? So we have total 3 bonds here: hydrogen, peptide, disulfide. Disulfide and peptide are covalent and hydrogen bond is non-covalent. Other than this, what are seen as extra in the tertiary and the quaternary group? That I will be writing it here, okay? So these are the list of bonds. The total we have in proteins, the 6 type of interactions or chemical bonds. Chemical bonds are the ones mainly which are having the strong ability for holding the molecules. We call them as bonds, and other are said to be simply the interaction here. So look at these type of chemical interactions in the tertiary and the quaternary structure. So we have the peptide bond, the disulfide bond, the hydrogen bond. Other than these 3, we have ionic bond, hydrophobic interactions and van der Waal interaction. We have discussed with the peptide bond, which is a bond between the C and N. We have discussed with the disulfide bond, which is the bond between 2 sulfur atoms, S and S. We have discussed with the hydrogen bond. Hydrogen bond is a bond between hydrogen and higher electronegative atoms like nitrogen and oxygen. And next we have the ionic bond. These are mainly formed by weakly sharing electrons. So we have some less electronegative atoms. So mainly they will be having the weakly sharing. You will be learning all these type of bonds in your chemistry, children. Okay, so coming to the ionic bonds here. Ionic bonds are the ones which are having the sharing of the electron pair, but they are weakly shared between these molecules or atoms, we call such bond as the ionic bond. Compared to the covalent bonds, the electron sharing it is very strong. Why? Because it is having some electronegative atom on one side and electropositive atom will be on the other side, which will make them very strong. But in the case of ionic bond, they will be having the weak interaction based upon the nature of the molecules, that is weakly electronegative or weakly electropositive. Okay, so they will be having the weak sharing bond between them. And next we have hydrophobic interaction. These hydrophobic interactions are very important. Why? Because generally the water is surplus inside the systems, and these water, they will form lattices or cage like structures around the molecules. Wherever there are polar amino acids, mainly they will form the cages or lattices. It will be very disturbing for these molecules to be present in their conformation. So in order to avoid these kind of lattices formed by the polar interaction, the non-polar amino acids are helping. Mainly the hydrophobic nature, it is present for the non-polar amino acid. Why? Because they have the hydrocarbon chains. These amino acids, they mainly help in the breaking of these lattices. So wherever the water cages or water crystals are formed around the amino acids, because of the presence of a hydrophobic amino acid, that will form a hydrophobic interaction. So it will repel the water molecule, so that will break the lattice or any kind of cage that is created around the amino acid chains. Okay, so this is very important in mainly in the repulsion, or you can say the removal of the cage structures around the amino acid chains. That will create a lot of wastage of energy, or entropy will be released. That is again a wasteful process. So in order to help in breaking these lattices, the hydrophobic interactions are helpful. So these are even seen in the case of tertiary and the quaternary structures. As these tertiary and quaternary structures are very closely placed structures, these extra interactions are seen like ionic bonds, hydrophobic interactions, and lastly we have van der Waals interaction. What is van der Waals interaction? Weak electron clouds are generally present. So there are different kind of charges that are displayed on each amino acid. Each will be showing impact on the other amino acid. So there will be some influence between two different kind of amino acids. Mainly with the charged amino acid, there are some electron clouds, temporary electron clouds that are created, which will influence each other, and such interactions are said to be the van der Waals interactions. So these are the weakest interactions which are present. They are not forming any bond, just they will influence each molecule. Okay, so they are generally influenced by each other. That is said to be the van der Waals interaction. So these are the list of interactions and the bonds that are present in the tertiary and the quaternary structure. So this is regarding the interactions and the chemical bond formation in the protein molecules along with their structures. So I have explained today regarding the protein structure. Let's see once again, children. The primary structure, linear chain having peptide bond. Secondary structure, alpha helix, beta pleated sheet. And here we have the peptide bond, hydrogen bond, and disulfide bond. In hydrogen we have intra and inter, but for alpha helix we have only intra, and for beta pleated we have both inter as well as intra. And coming to the tertiary structure, mainly the biologically active proteins are in tertiary or quaternary form. The tertiary means mainly the simple protein molecules made up of single polypeptide, and these are, they can form, the overall structure can be of two types. Either they are circular, which are called globular proteins, or they can be rod shaped, which are said to be the fibroid proteins. Here I have given some examples. And lastly we have the quaternary structure. The quaternary structure is again a biologically active protein molecules, but are for macromolecules. They are made up of many amino acids, thousands of amino acids, millions of amino acids are linked here. All these polypeptides, in this case only one polypeptide, it will be having different structures. The beta pleated, alpha helix, this is conformation, but here it is, we are talking with the chain of the single chain which is present in alpha helix or beta pleat or in combination. But in this case, the quaternary structure, we have the different polypeptides with the different units of alpha helix, helices and the beta motifs. Okay, so different polypeptides, they will come together to form a single protein, and they will show numerous interaction, and such type of structure of the protein is said to be the quaternary structure. Here I have given examples, hemoglobin and myoglobin. And essentially we are talking with the interactions here. We have peptide bond, disulfide bond, hydrogen bond, ionic bond, hydrophobic interactions, and van der Waals interaction. Okay, so this is regarding the structure of the proteins and the chemical bonds along with their interaction. Now let us see overall strength of these chemical bonds. So look at the order of the strength of the chemical bonds and interactions I have given. This is very important, children. So even in your research, it is very important to know the type of interactions, how these structures are mainly formed. So this is very important chapter and very important section where you have to pay a lot of attention. Okay, so mainly if you see the interactions here in order I have given, the peptide bond is strongest bond. Why? Because it's a covalent bond. The first two bonds will come under the covalent, and all the remaining interactions and the bonds are said to be the non-covalent interactions. Okay, these are mainly formed by the sharing of electrons between a highly electropositive and a electronegative atom, and hence they are said to be the strongest bonds. So here look into the order of the chemical bonds here. So peptide bond stronger than the disulfide bond. Disulfide bond is further stronger than hydrophobic. Hydrophobic is more compared with the ionic. The strength of ionic bond, and ionic, it is greater compared to the hydrogen bond, and hydrogen bond, it is again greater with the van der Waals interaction. So compared to all these bonds, the peptide bond is said to be the strongest bond, and the weakest bond is said to be the van der Waals bond. Okay, so this is the order of the chemical bond, the strength of the chemical bond. This is even very important for the other exams. For NEET it is not required, but for other exams like AIMS and ZIPMER, it is very important, children. Okay, so this is about the chemical structure. This is about the structure of the proteins, their conformations, and the type of chemical bonds and their interaction. In the next lecture, I will be meeting you with the new topic regarding the structure of proteins, and even I will be giving some introduction regarding the lipids.